In this presentation, I'm going to talk to you about how we can view the effects of different antibiotics on bacterial growth. So to begin this procedure, we need petri dishes, and those petri dishes are filled with an agar, a jello-like substance which contains nutrients. The next step is to cover the petri dish with a sample of bacteria. So the way we would do that is we would get a sample of our bacterium on a swab, and we would swab over the agar and kind of try and cover that with bacteria. And if you've taken or will take microbiology, there's a particular way that you cover the petri dish with a colony of bacteria. So we kind of swab the petri dish or the agar with that bacterial sample. So the idea is to cover the area and allow that bacteria to grow and kind of fill in all of these spaces. So what the purpose of this experiment is to see how this bacterial growth is going to be affected by different antibiotics. So we have a single species of bacteria that we've covered the petri dish with, and then the next step is to add some antibiotic discs. Basically, the antibiotic discs are pieces of paper that have been soaked in an antibiotic. And so in this experiment, what we did was add 4 antibiotic discs, and they stick to the petri dish. So each of these 4 circles represents a different antibiotic. And then what we do is place this petri dish with those antibiotic discs in an incubator for 24 to 48 hours and allow that bacteria to grow. So what I'm going to show you next are the results of those petri dishes. So this first petri dish shows how Staphylococcus epidermidis grew in the presence of these 4 different antibiotic discs. So here you can see the bacterial growth. It's this kind of tan colored sweeping marks all over the agar, and then each of these discs again is a different kind of antibiotic. And what we would do is, for example, down here you can see this CYP5. That's an abbreviation for ciprofloxacin, which is a kind of antibiotic. And what we would do, we would measure the diameter of this dark area right here. And this dark area around that antibiotic disc is referred to as the zone of inhibition. So basically we covered the petri dish with bacteria. The bacteria would normally grow all over the place. However, if that particular bacterium is affected or inhibited by a particular antibiotic, it's not going to grow in that area where the antibiotic is present. So this is showing us that Staphylococcus epidermidis is susceptible to the antibiotic ciprofloxacin. You can see down here we have another antibiotic that has a smaller zone of inhibition but still shows growth inhibition of the Staph epidermidis. These 2 antibiotics have no zone of inhibition, and therefore Staphylococcus epidermidis is unaffected by those 2 particular antibiotics. So let's look at another example. This is showing you Bacillus subtilis, right? So the Bacillus subtilis was spread all over this agar to try and grow, and then those same 4 antibiotic discs were added to the petri dish as the bacteria grew in the incubator. And what you can see is that Bacillus subtilis is susceptible to all 4 of these different kinds of antibiotics. So the zone of inhibition is relatively large for each of these 4 antibiotics that are shown. So here's the last example. This is showing you E. coli grown in the presence of those same 4 antibiotics, and it's unaffected by 2 of these, and you can kind of see a faint zone of inhibition with these 2 antibiotics. So again, E. coli is affected by 2 and unaffected by 2. What you'll notice from these pictures is that different bacteria are affected by different antibiotics, and that's kind of important when we think about treating diseases with antibiotics and knowing which kind of bacterium an individual has an infection with, and therefore knowing which kind of antibiotic should be prescribed to treat that infection.